So, I took a look at how you all did on the pre-class quiz. Overall, did very well, right? So I do want to go over some time, just at the beginning, going over a little bit of protein structure. What I saw a lot of people struggle with actually was the secondary structure. Helices, beta sheets, beta strands, things like that. So I'll spend some extra time on that. And I'll open up for questions, either from the chapter or from the homework. I have an example problem that I want to go over, and then we'll get into our case study, hopefully. Right? So first, what makes up a protein? Well, protein is one of those macromolecules that we were talking about on Tuesday. The macromolecule, in this case, is biopolymer. This polymer made out of amino acids. We're going to go over amino acids in just a second here. Right? Now, when we talk about a protein, really what we're talking about is a polypeptide. Right? In this case, the polymer is being held together by peptide bonds. So each amino acid is held to the next amino acid by a peptide bond. And this peptide bond has some interesting properties that we'll see in just a minute. Now these polymers, right, once we have, whether it's 50, 100, 500, or 1,000 amino acids all held together by a peptide bond, they have to fold, right? So when we talk about protein structure, really what we're talking about is a protein fold. Into what structure does this linear chain of amino acids fold itself, right? And we have 4 orders of organization in our protein structure. We have primary, which is simply just the order of amino acids in which they're added. Right? So we start at the N terminus and we end at the C terminus of the protein. We'll see, just like DNA, that proteins have polarity. They start at one end and end at another. They're asymmetric. Then we have our secondary structure. Our secondary structure, now spend a little bit more time going over, our alpha helices and things like that. These are stabilized by hydrogen bonding. Now, once we have our secondary structural elements, these secondary structural elements can interact with each other via other weak interactions like hydrogen bonding, or most predominantly the hydrophobic effect. And these put together a fully functional protein some of the time. This is our tertiary structure. Secondary structural elements interact. At this point, we might have a fully functional protein or enzyme. That's not always the case, though, because there's a 4th level of organization that some proteins or enzymes exhibit. This is the quaternary structure. The quaternary structure, again, is optional. Not every protein has a quaternary structure. But for example, here we're looking at is a DNA beta clade, right? So this is, we're going to see when we talk about DNA replication, this forms around the DNA. It holds the DNA polymerase on the DNA. It's made up of 2 subunits, right? So each of these subunits, the red and the yellow here, are a fully folded polypeptide, right? So they've already adopted their primary, secondary, and tertiary structures, but on their own, they're useless. It's only when we get 2 polypeptide chains coming together and interacting in the quaternary structure that we get this hole in the middle here and allows the clamp to form around DNA, right? We can see the same thing when we talk about hemoglobin in the next chapter. When we talk about hemoglobin has a quaternary structure. It's made up of 4 subunits. Any of the subunits on their own is useless. It's only when the 4 subunits or 4 polypeptide chains come together and interact and form a fully tetrameric hemoglobin that we get a functional oxygen carrier. Now, proteins have different properties, right? So as we'll see, proteins are made up of 20 amino acids. You should have seen this in the text, right? So there are 20 alpha amino acids that make up all of our proteins. That's not to say that there are only 20 alpha amino acids that exist. In fact, when we get to the urea cycle, we're going to see 2 or 3 new amino acids that are not part of proteins. So in this case, there are 20 specific amino acids, or alpha amino acids, that make up a protein. Okay? Now, these 20 amino acids have varying properties, right? We have 4 different types. We have nonpolar, and we have polar neutral, polar positively charged, and polar negatively charged. By extension, because these make up a protein can also have different properties. It can be polar on the outside or non-polar on the outside, or it can be charged, uncharged, and have different isoelectric points like we'll talk about in just a minute here. Okay? Some proteins can also be rigid, right? So they have very little flexibility. Some proteins are more flexible. This is important. So a cytoskeletal protein you would expect to be more rigid. It's giving the cell shape. You wouldn't want that to be flopping all around. Other proteins, like we have here, ferritaxin, which binds iron fact, many proteins that interact with their substrates will bind around that substrate. We'll see an example of this when we get to the citric acid cycle in citrate synthase, right? So proteins can have many different properties. They can look very different ways, but they all have the same same levels of organization: primary, secondary, tertiary, and sometimes ordinary. Right? It just depends on the protein. Now, if we look at an amino acid, an alpha amino acid, they have a few things that are always in common. All alpha amino acids are going to have an alpha carbon in the center. This is the hub of our alpha amino acid. And to this C alpha, we're going to have always connected a carboxyl group, an amino group. We call this the alpha amino and the alpha carboxyl. We're also going to have a hydrogen and this R group. This R group is the only thing that varies among those 20 amino acids. Otherwise, they're exactly the same, with the exception of maybe proline, right? But otherwise, they're exactly the same. And they're always found in the L configuration, right? If you notice, that's C alpha, okay? We just consider R to be some hypothetical thing. Well, now this C alpha is connected to 4 different and unique substituents, which means that it's chiral. This C alpha is chiral, meaning that we can have either the D stereoisomer or the L stereoisomer. As it happens, evolution has selected that the L isomer, the L stereoisomer, is the only stereoisomer that's going to be used in our proteins. Now, all amino acids also have at least two ionizable side groups: the alpha amino group and the alpha carboxyl group. The alpha carboxyl group, the proton on here is ionizable, the pKa of usually between 8 and 9. The carboxyl can exist as either COO minus or COOH, right? So that's got an ionizable proton as well, with a pKa typically around 2 or 3, right? So let's examine this carboxyl here. At biological neutral, which is pH 7.4, is that carboxyl going to be protonated or deprotonated? You're right there. Is it going to be protonated or deprotonated? The carboxyl here at pH 7.4. Is it going to have the proton or no? Huh? Deprotonated, exactly. The pH 7.4 is higher than the pKa, so the proton is gone. Now, what about the amino group? Is that going to be protonated or deprotonated? Deprotonated, right? The pH is lower than the pKa. So at biological neutral, any amino acid is going to exist as what we call a Zwitter ion, right? Meaning that it is ionic, it's got charges on it, but the overall charge on the molecule is 0. So when we draw an amino acid, we always are representing it at biological neutral, so we always depict it as CH3 plus and COO minus. If the pH changes, that's the example I have here, that's what we've done, right? Well, now we have to consider, right, what is going to be protonated and what is deprotonated. All right, now we can break down our amino acids into these 4 categories. I won't spend too much time here, right? But here we have the hydrophobic amino acids, the nonpolar amino acids. Of note here is proline. We'll see proline again in just a second here. But notice proline here is the only amino acid where the R group loops back over and connects to the alpha amino group. Right? All other amino acids just have their R group sticking out. Proline is the only one that loops back around and actually connects to the backbone, right? This alpha amino group. We have our polar neutral amino acids. So these have polar functional groups, but they have no charge. So here we have some important ones: serine, threonine, and tyrosine, which we'll talk more about in just a minute, and asparagine. So these 4 are critical for any for any protein. We also have glutamine, cysteine, and histidine, right? So histidine also important. We'll get to that. I have a slide here with all of the notable amino acids. We have our positively charged, our polar positively charged, and lysine and arginine, and we have our negatively charged lysine. Negatively charged should be aspartate and glutamate. Sorry, I'll fix that on the slide. I don't know why. All right, so negatively charged should be aspartate and glutamate over here. So sorry about that. Okay. So now we have our 4 categories of amino acids, and notable among them are these, right? So serine is absolutely critical for enzyme regulation. protein regulation in general, because serine can be the site of either glycosylation or phosphorylation. We come back here to serine, you see on the top left here, serine, the R group, here's our alpha amino, alpha carbon, alpha carboxyl. The R group ends in an OH. This OH can attack an orthophosphate or can participate in the group transfer reaction from ATP, such that this carbon, the beta carbon now, this one right here, right, this can hold on to a phosphate group instead of a hydroxyl. We call that phosphoserine. Serine can also hold on to sugar modifications by a glycosidic bond. We'll see that in chapter 11. Phosphorylation and glycosylation are two major ways that protein activity or enzyme activity is regulated. Phosphorylation very often acts as a switch, right? If a protein is phosphorylated at a certain serine residue, then it's on or off or whatever, right? So serines are important. Threonine also ends in a hydroxyl, so we can see over here, this hydroxyl on threonine can also harbor a phosphate group, so we can have phosphothreonine, and the same is true for tyrosine. Right? Serine is by far the most common amino acid that's phosphorylated, but threonine and tyrosine can also be phosphorylated. Asparagine is important because asparagine can also be the site of glycosylation, just like serine. You notice serine is the only one really that can be either, right? But we can have phosphothreonine, phosphotyrosine, or asparagine glycosylated. And again, glycosylation serves as a regulator of enzyme activity, right? Think about it, that makes sense. If you think about metabolism, well, certain enzymes in metabolism we want to be switched on when sugar levels are high, right? And that's what glycosylation is. It's a sugar modification. So if sugar levels are high, then we might expect a higher level of glycosylated asparagine, which would do something to the activity of that specific metabolic enzyme, either turning it on or off in response to sugar. We have cysteine, which is also critical. Cysteine is able to form covalent disulfide bridges in its oxidized state, right? So when we have two cysteines that are oxidized next to each other, they can form a disulfide bridge. We go back and look at the structure of serine, we see this sulfhydryl here. This, what we're looking at it, is in its reduced state. We can oxidize the sulfhydryl, and we have an extra electron. Okay? If there's another oxidized sulfhydryl nearby, well, now they can share one electron from one sulfhydryl, the other electron from the other, and now we're sharing a pair of electrons, which is a covalent bond, linked together by two sulfurs. This is our disulfide bridge. This helps to either stabilize the structure of a protein, locks it in the correct fold, or it can actually help two interacting proteins, right? So we can have a covalent link between two interacting proteins. We can lock them in place. We have proline, again, we mentioned proline before. This is the only amino acid where the functional group, the R group, loops back around onto the backbone of the amino acid. And this constricts its kind of freedom of movement, right? As we'll see in just a minute. Okay, amino acids are free to move within their alpha amino and C alpha bonds, and their C alpha bonds and alpha carboxyl bonds, right? So these are free to twist and define the psi angles. Histidine, all right, we have an R group with pKa6, right? And this is the only amino acid with a ionizable side group with a pKa of very close to neutral. So we find, I mentioned this on Tuesday, we find histidine very often in the active sites of enzymes. We're going to see this when we talk about hemoglobin. It can serve as either a hydrogen bond donor or not a hydrogen bond participant, right? It can also serve as a proton donor in some cases. Okay? Now here, histidine is an example of an amino acid with an ionizable side group, but there are actually 7 amino acids that have ionizable side groups. All 20 are going to have the ionizable alpha amino group and the ionizable alpha carboxyl group, but 7 of them also have an ionizable proton in the R group, in the functional group. And those are aspartate and glutamate, right? These have a pKa of about 4. Histidine, again, a pKa of about 6. Cysteine, pKa of 8.3. Tyrosine, 10. Lysine, 10. Arginine, 12.5. So we see that we have 4 basic amino acids, and we have, let's call it 2 acidic amino acids. And let's just call histidine neutral, right, because it's so close to biological charge of our protein is and what its isoelectric point is, okay, depending on what all of these are. Now, when we talk about amino acids, we never talk about amino acids in their full names, right? It's just too arduous. If you're going to write down the sequence of a polypeptide, you don't have to write down all the names. So we refer to them by their single letter codes. This is one thing that I have to insist that you do memorize, right? You should know this. Now, some of them make sense, like A for alanine or G for glycine. Some of them don't make sense, like K for lysine or R for arginine, right? The ones I remember, you know, the weird ones are R for arginines, like you're saying arginine, like a pirate, like arginine, or tryptophan is W. You pretend like you say tryptophan like a baby talk, tryptophan, right? Anyway, all right, so let's move on here to the different orders of structure. So we have primary, secondary, tertiary, and quaternary. Seems like primary, very straightforward, right? So most people did very well on this part of the quiz. Just to go over again, we have 2 amino acids, right? And what we have here is the formation of a peptide bond that we see here with a release of a quadrant, right? 2 hydrogens from the alpha amino group of 1 amino acid and the oxygen from another amino acid. The alpha carboxyl group is binding to the alpha amino group, so we get a bond in between the amino acids. Because we release a water, this is a condensation reaction. So once we have an amino acid inside of a polypeptide chain, that amino acid is now always referred to as a residue, right? So when we talk about a serine residue or a glycine residue, that's just a glycine in the context of a polypeptide chain. Because the way that it got there was a condensation reaction. What's left over after condensation, of course, is a residue, right? So if we look at a longer polypeptide that we have here highlighted in yellow, okay, well we have NC-alpha-CO. Here's our tyrosine, right? So we have tyrosine here. Here we have NC-alpha-CO. Here's a glycine. Here is a hydrogen. NC-alpha-CO. Here again, glycine. Here we have NC-alpha-CO, alanine. So notice the only thing that's changing is the identity of the functional group. And if we look at this entire polypeptide, we see that at one end we have a free amino group, and at the other end we have a free carboxyl group, right? This gives a polypeptide polarity. Just like DNA has a polarity of 5' to 3', a polypeptide has a polarity from the N-terminus to the C-terminus. And what we see here is just repeating always, right? So this is what we call the backbone. NC-alpha-CO, 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 and so on ad infinitum. So if I give you, for example, a tripeptide and tell you to identify, right, the 3 amino acids that make it up, what's the first thing that you should look for? Huh? Before that, before the R groups. Huh? The N-terminus, sure, and then the C-terminus, but that might be confusing us. We'll see in just a minute. Right, so just looking for the N-terminus and the C-terminus might be confusing. Well, if we see here, one thing that all polypeptides are always going to have in common, regardless of the identity of the amino acids, is that there will always be a backbone with the same repeating pattern, NC-alpha-CO, 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 on and on. So the first thing that you should do to identify a polypeptide is to look for the backbone. Once you have the backbone, now you can see where the C-alphas are, and now you can look for the R groups, right? As you'll see again, just looking for an amino group or looking for a carboxyl can get you into trouble because some amino acids have amino groups in their side chain and some have carboxyls in their side chain. So the first thing you want to do is look at the backbone. Now when we look at this here, of course, now we have, like we said, tyrosine, glycine, alanine, and leucine, right? We're going to want to write it like this, right? So again, memorize those single letter codes. What would we write this as? Tyrosine is Y, glycine is G, phenylalanine is F. You just pretend that you're illiterate, right? Phenylalanine. And then leucine is L. So what do we have here? We have YGGFL. So with those 5 letters, YGGFL, we're able to recreate this entire structure. And one thing that we're going to see over and over this semester is that biochemists are very lazy. So they don't want to have to write down the structure every time. If we can do the same thing with 5 letters as we can do with this monstrous looking thing here, we're going to do that. Okay? So memorize those 1-letter codes. Now again, those cysteines are important because cysteines can form these disulfide bridges. close next to each other that are oxidized, right? Oxidized. Now, they're each losing one electron, so we get 2 electrons lost. They're sharing a pair of electrons, but we also get 2 protons, right? So effectively, what we've done is we've lost 2 hydrogens here. Now they're sharing a pair of electrons, and we get a covariant link between them. This can be intramolecular, so an intrachain disulfide bridge. We're going to see this in the Anfinson experiment. Holds the protein together, it blocks it in the correct conformation, or we can get it in between 2 different polypeptide chains that we see here. We see examples of both. What we're looking at here is insulin. Insulin is made up of 2 chains. We have an A chain and a B chain, and both are just short polypeptides, right? Nothing magical about insulin. This is what insulin just is, right? Whereas we notice we have an intramolecular or an intrachain disulfide bridge in the A chain, right? So this cysteine and this cysteine over here are linked together, so that's holding the A chain in the correct configuration. But we need both of these to be together. The A chain on its own is useless. The B chain on its own is useless. It's only when they're together. So how do we ensure that these 2 short polypeptides are always together so they form a functional insulin? Well, we can't rely on weak interactions in this case because these polypeptides are too short. There simply aren't enough weak interactions possible to hold these 2 together. Additionally, insulin, where do we find insulin? Huh? In the blood, right? So it's made in the pancreas, sure, but then it's a hormone, right? The blood, right? So the lymph system, they're actually very hostile environment. So things that have to be together outside the cell, we typically find with disulfide bridges, right? So in this case, we want to make sure that the A chain and the B chain are always together. So we put disulfide bridges together.
If the A chain stands alone, not connected to the B chain, will that extracellular, will that like disulfide bridge still happen? The one that's only on the A chain?
Sure, right? So all we need for this disulfide bridge to occur is for this cysteine and this cysteine to be near each other and be oxidized. So even if we ignore the B chain, that can still be possible, right? Good question. Okay, so let's move on here. Peptide bonds, right? So holding these together. We already talked about how the peptide bond forms, but what I want to bring home here is that peptide bonds are almost always in the trans, well, they're always in the trans configuration, right? And what do I mean by trans? Well, if we look, well, the trans can only happen, right, if we have a double bond. And that is possible because peptide bonds have this resonance structure, okay? So this is how we typically draw a peptide bond, okay? But this is also as equally correct, meaning that a peptide bond has partial double bond characteristics, right? And what we find is that the peptide bond between 2, generic amino acids is always going to be trans, except for proline, right? The reason being, here in the trans configuration, there's less steric hindrance between the carbonyl oxygen and the amino hydrogen here, right? So this is favorable. If we now make it cis, well, now we're getting steric hindrance between the R groups of neighboring amino acids, right? So this is not favorable. So we always find it in the trans configuration. Now this is not the case for proline, right? So we have X, which is any amino acid followed by proline. What we see is that we get just about equal steric hindrance whether proline is in the trans configuration or in the cis configuration. So proline is the only one that can exist in the cis configuration, not because it's more stable, but just because it's as stable as the trans configuration. Now secondary structure. This is where everyone had problems on the quiz, right? So what was the issues that you had with secondary structure? What wasn't clear about secondary structure? So we can focus on that. Raise your hand if you understood what an alpha helix is. Raise your hand if you didn't understand what an alpha helix is. There's only 2 options. All right. So alpha helices are a type of secondary structural element. They're alpha helices because they were the first secondary structural element we discovered. Alpha helices look like this. Okay? And what this is a right-handed helix. Okay? Helices can be either right-handed or left-handed. In the case of proteins, alpha helices are always right-handed, with few exceptions that I don't want to get into. But for the most part, alpha helices are always right-handed. What do I mean by right-handed? Well, if you take your right hand, right, and and you point your thumb, right, you follow in the direction of your thumb, can you curl your right hand around the alpha helix, or around the helix? In this case, yes, right? If you try the same thing with your left hand, you can't. Okay? It doesn't matter if it's upside down or whatever. It's always going to be right-handed or left-handed. It doesn't matter which orientation you look at. Now that can be difficult with some people, the right-handed rule. Something else that helps is to imagine a helix be a spiral staircase, and you're standing at the top of the spiral staircase and you're walking down the staircase. If you're turning to the right, it's a right-handed helix. If you're turning to the left, it's a left-handed helix. So in this case, if we look at ourselves at the top of the helix there, and we walk down, we're turning to the right as we descend. So this is a right-handed helix. Now, if we look at the structure of an alpha helix, what we see is that we have an amino acid repeating every 3.6 residues, right? So it's got a 3.6 residue per turn and 3.6 residue pitch. Right? We start here, right? Or actually, let's just start right here and go down 360 degrees, right here. Okay? There are, on average, 3.6 residues. Now, of course, we can't have 0.6 of a residue, which means that it's just the residuals of that. In fact, if you look at this amino acid here, or this residue here, and go straight down, you see that there's nothing there, right? Instead, it's a little bit this way or a little bit that way, right? But the pitch is 3.6 residues, which means on average, if you had an amino acid of, 36 amino acids, an alpha helix of 36 amino acids, that will be just about 10 turns of the helix, right? So it's got that pitch. And that's always the case for a freestanding alpha helix. It's got a rise of 1.5 angstroms. It means that from this amino acid to this amino acid, the distance, the vertical distance between them is 1.5 angstroms. Okay, there's about a 10 degree or no, 10 degree, 100 degree turn between each of them, right? Now, we look here. How is an alpha helix formed? Well, an alpha helix is stabilized by hydrogen bonding between the backbone carbonyls and the backbone amino groups, right? So here, for example, we have a backbone carbonyl. We talked about this last time. This oxygen is a hydrogen bond what? It's an acceptor or donor? It's an acceptor, right? This oxygen forms a hydrogen bond with this hydrogen bond donor, right? And alpha aminos on a residue 4 amino acids away. 1, 2, 3, 4. Here we get a hydrogen bond between the backbone. Here we get a hydrogen bond between the backbone on the next carbonyl. Here we get a hydrogen bond between the amino group on the next one, and so on and so on. And we can see all those hydrogen bonds here in the dotted lines, right? So here's a volume stick representation of an alpha helix. It's stabilized by hydrogen bond between the backbone carbonyls and the backbone amino groups. Now, if we look at it top down and look at a space fill model, like we see on the bottom here, we see that the inside of an alpha helix is not hollow, as this implies. Instead, it's completely filled up by all of the backbone atoms. And what we see is that the R groups, the functional groups, are actually pointing out away from the axis of the double helix. And this gives the double helix some interesting properties. Depending on the sequence of amino acids making up that alpha helix, we can have, for example, all the amino acids on this side be polar and all the amino acids on this side be non-polar. And now we have an empty-packet helix. One half of it is polar, one half of it is non-polar. We find this very often in proteins that have to interact with, say, the phospholipid bilayer, right? Form some interface between the phospholipid bilayer and an aqueous pore in it, for example, right? So this is an alpha helix. And we can have, right, our primary structure of amino acids, right? So we have our N to C in this direction, right? So this group of amino acids here, right? Let's say that they have the correct propensity to form an alpha helix. So we say, all right, there's an alpha helix there. Over here, there'd be another alpha helix. Let's say over here, there's another alpha helix, right? If an alpha helix forms is down to purely the sequence of residues, right? So if these residues were changed to something else, there's nothing to say in this particular protein that this particular region has to be an alpha helix. No, that forms spontaneous through thermodynamics. The lowest energy state of that sequence of amino acids in that region. Why? Well, it's because all of these hydrogen bonds are forming. Okay, all these hydrogen bonds are forming, releasing energy. It's the lowest energy state. So the most stable configuration for this region to be in is an alpha helix. And the same over here, and the same over here. Okay? Now, proline, glycine, and branched chain amino acids are not allowed in, what do you call it? In alpha helices, right? And why is that? Well, proline, again, remember, is constrained. We go back to the structure of proline, we'll see that's the one that has that R group that loops back on the alpha amino group. So here, proline at the top there, okay? This bond angle between the alpha amino group and the alpha carbon is constrained by this loop, so it can't twist, and it can't adopt the correct angle in order to be part of an alpha helix, right? So that's just simply not allowed. Glycine is similarly not allowed, okay? Because, well, actually not similarly, for the opposite reason. It's too free. Notice that the R group on a glycine is a hydrogen, right? We have the hydrogen that we normally find on the alpha carbon. We also have a free hydrogen, okay? That's our R group, so this is conformationally unrestricted, and so it destabilizes the helix, right? This bond angle between N and C is too free. And the branched chain amino acids, like isoleucine and we have leucine and valine, right? These are also not allowed because they're too big and they hinder the formation of the hydrogen bonds that stabilize the alpha helix. So without those hydrogen bonds to stabilize the alpha helix, well, now this region of amino acids no longer will form an alpha helix because that's now no longer the lowest energy state, right? That big, bulky hydrophobic group is getting in the way. Now, beta sheets, right, are a little bit easier to understand. You can think of them as just an elongated series of amino acids. And beta sheets are made up of beta strands. Okay, so beta strand, you see here, has a length of 3.5 angstroms per residue, right? And interestingly, if we look at just this beta strand, okay, let's just look at this beta strand right now. Notice that here, the R group is facing towards us. Here, the R group is facing away from us. Here, the R group is facing towards us, away from us, towards us. So we have this alternating pattern of where the R group is. And this gives beta strands the same or similar property of what we saw in amphipathic helix. We can have one side of the beta strand that is nonpolar. We can have one side that is polar. Right? So we also find beta strands, amphipathic beta strands, again, at the interface of between a nonpolar and polar environment. Like, for example, thorium in the phospholipid bilayer. Now these beta strands can hydrogen bond with each other to form beta sheets. We can have either an anti-parallel arrangement, where we have N to C going in this direction, and then N to C going in the opposite direction of the next one, right? Or we can have a parallel arrangement. We have N to C going the same for both directions. So we can have either purely anti-parallel, where we have however many strands we have, there's no limit, right? Say that we have 5 beta strands making up a hypothetical beta sheet. This is purely anti-parallel. Or we could have them all going in the same direction, which would be a parallel beta sheet. Or we can have, like we see here, a mixed beta sheet, which is by far the most common. It's very rare that we see purely anti-parallel or purely parallel beta sheet, right? Most often it's mixed. But similarly to alpha helices, a beta sheet, which is made up of these beta strands, is stabilized by hydrogen bonding. Okay, the strands hydrogen bond with each other, right? So here we see some arrangements of beta sheets, right? Beta sheets are a lot more flexible in terms of the configurations they can adopt, right? We can have just a flat beta sheet, we can have a saddle-shaped beta sheet, we can have a barrel-shaped beta sheet, okay? These are all allowable, right? The reason is, okay, we don't have to adopt specific bond angles to get that periodicity that we get in an alpha helix. Instead, all we have here is just an elongated stretch of amino acids. So they can be a little bit bendy, they can be a lot of bit bendy, like you see over here, for example, right? This is pretty bendy. So this allows beta strands to come together to form beta sheets in many, many different types of configurations. Here's an example of only 3. Okay, so let's say that now, okay, here in this region, this forms a beta strand, and this forms a beta strand, right? And maybe over here we have another beta strand. These can all come together to now form a beta sheet, right? But we can't get there yet because all we've done is now we have this, linear thing of amino acids, okay? How do we get all of these elements near each other? Huh? Well, that can be accomplished by turns and loops, right? So turns and loops are not like the name implies, just nebulous turns and loops all over the place. Instead, they're structured. Okay, here we have a beta turn, right, which is made up of 4 amino acids, exactly 4 amino acids, and it's stabilized again by hydrogen bonding, right? So we would get, for example, a beta turn, right? We might say that we have a beta turn right here. So we get these 2 beta strands bending down and now forming an anti-parallel beta sheet, okay? And it's stabilized by hydrogen bonding between the first amino acid of the beta turn and the last alpha amino of the beta turn. And we can also have beta loops, right? Beta loops are actually very interesting. Beta loops are what's responsible for antibodies being able to recognize different antigens. If we look at them here, okay, well, they look like they're just here. Of course, we have beta strands, and they always point in the N to C direction. So here's the C terminus of this beta strand, here's the N terminus of this beta strand, on and on. And now notice here, this is connected by a beta loop to this beta strand here. Okay, this is not just amino acids all floating around. Instead, beta loops do adopt a defined structure. They're again stabilized by weak forces, right? So now we have alpha helices, we have beta strands, and now we have the beta loops and the beta turns that can twist this entire thing, right? And how do they twist? Well, that's the tertiary structure of proteins. It's when these secondary structural elements, right? Alpha helices, beta strands, beta sheets, beta turns, beta loops, when they all interact with each other, again, via weak forces, notably the hydrophobic effect, to form the lowest energy state, right? So we see here is myoglobin, right? So myoglobin looks like this. So what we see here is the tertiary structure for myoglobin. We see we have 7 alpha helices here. We have a heme group, which we call a prosthetic group. We'll talk more about that when we talk about myoglobin in more detail and hemoglobin. But for now, just know that there is a heme there. But notice how all of these alpha helices are interacting with each other, right? And oriented in very specific ways, right? That is the tertiary structure of a protein. And in the case of myoglobin, because it's monomeric, meaning that it doesn't interact with any other protein partner, we're done, right? It's adopted the tertiary structure, and it's a fully functional enzyme, all right? Here we see the space-filling model, right? So we see that while this is nice because it allows us to see what the secondary structural elements are, proteins don't actually look like this. Instead, they look more globular, right? That's an actual scientific term somehow, globular, right? So that's what we see. And if we take a cross-section on the bottom, that's what we're looking at is the cross-section. Here, the yellow amino acids, or the yellow residues, are hydrophobic, non-polar. The blue amino acids are polar, right? We take a cross-section of that, so we can cut this structure in half. What we see is that the inside of myoglobin is mostly non-polar, mostly hydrophobic, and the exterior is mostly hydrophilic or polar, right? This is what mainly drives the formation of tertiary structure. Yes, there's weak interactions between the secondary structural elements, but for the most part, what we see is that hydrophobic effect, like we talked about on Tuesday, forces the non-polar residues to the interior of the protein, leaving the polar residues on the exterior of the protein. Why would we want to do that? Because they're found in the cell, which is mostly water, right? So we want the polar residues on the outside so that that can remain soluble. We can have, though, inside-out proteins, where it's non-polar on the outside and polar on the inside. Where might we find a protein like that? More generally. In the membrane, right? Remember, the membrane is made up of that phospholipid bilayer. We're going to talk about that in chapter 12 or 13, something like that, right? Now, this isn't the end of the story for most proteins. Most proteins also adopt a tertiary structure. Like, for example, hemoglobin. I already talked about hemoglobin, so I won't stay there. In that case, the tertiary structure is not optional in the sense that a protein that has a tertiary structure doesn't have to adopt the tertiary structure. But that if a protein does have the tertiary structure, it must adopt the tertiary structure. Ordinary structure. Yeah, ordinary structure, right? Like hemoglobin, what we're seeing here. So again, 2 alpha chains and 2 beta chains is a tetramer. Okay, so they have to be interacting in order to be functional. Any one of the chains on their own, right, are useless. So if a protein has a quaternary structure, that quaternary structure must be achieved in order for that particular enzyme, protein, whatever, to be functional. Okay? So let's pause there. Is anything not clear about secondary structure? Ask your questions now. It's the whole point of this whole part of class.